Everybody, my name is Gunnar Red, and I'm happy to introduce the next speaker. So our next talk is by Christopher Mason. He's from the Weill Cornell Medical College in New York City. Chris is very much involved in the COVID-19 response and research in New York. It's great that we can get him on short notice and for some time here to share some data and some stories and some results related to COVID-19 in New York City and around the world. I think it's fair to say that Chris is a technologist and he's driving and is being driven by technology, specifically on RNA and DNA sequencing technologies. So today he's going to talk about his recent efforts in global mapping of RNA viruses, their hosts and their variants. Chris, I'm very glad you could make it. I'm really looking very much forward to your talk. So let me unmute. Chris, are you there?
Okay, great. Yes. Okay. Can you hear me okay?
Yes. We don't see your slides yet.
Yeah, so good morning and good afternoon, everyone. Thanks for having me. It's a pleasure to be here. As most of you know, we're in the middle of sort of the epicenter of the crisis in the US, and so I haven't been sleeping much. So if I sound sleepy, it's because I am sleepy. But let me share my screen here and go over some of the work that we're doing, and also some exciting new results I think that'll help everyone on some ideas and planning. So let me get this up here. So, I'm gonna go, let me see if I can minimize this and you can see my screen, right? Make sure it looks okay. Great. So I'll keep it brief. It's really just about a 10-minute talk just to go over some things we're doing in the lab and elsewhere around the city, and also from a global perspective, what we've been doing to look at the question of where the viruses might live elsewhere outside the body. So I'll start, obviously with the background. Most people know this, this map. If you haven't been seeing it, it's a really great but yet terrifying map of where the cases are. And so things have really spiked here in the US recently. And you can see here there's many cases will almost certainly surpass 1 million probably within the next couple of days. So it's not news to anyone. But what we've been doing is both in the clinical samples and also in environmental sampling, this idea of can we see how many of the viruses and evidence of them in the environment, and then compare that to what we're finding in patients. This is kind of building off a consortium of which Gunnar and I have been working on for years. Actually, is a consortium of metagenomics and of researchers looking at subways in urban biomes as metasub. It's basically, we started sampling on March 16th to look in public surfaces and hospitals to really address the question empirically of is the virus surviving on surfaces. There's been a few papers looking at hospitals and other environments to see that they can sometimes get a virus that's there, and sometimes even infectious virus, which are two important distinct sample types. But we wanted to build this as a global map and see this as the pandemic progressed, what can we observe of what's present in the cities, and then also what we find in patients. So in particular, this work, Andre Callas, so I don't know if he's on the call, but has also been building basically a global KMER index, some of which you can browse at this website below to see what we see. We've been building this at the DNA side, and in the past few months, we started to build this index out also for the RNA side, with the idea that if we had been doing this routinely, we might have been able to actually track the emergence of different RNA viruses, as we already can for DNA viruses before it would have become potentially problematic. And so this is an infrastructure that we've been building on for years to begin this mapping. I'll share some of the results with you today. But this is kind of the concept and a very nice figure that Andre made to show this sort of concept. So there's a multi-sample sequence index, and you can search actually on this website below. Even if you don't know what a piece of DNA is, you can at least tell where it's being seen, which is something we'll end up need to be doing for other viruses that are constantly emerging. And also, since I know it's a machine learning group, we wanted to tell you one other bit of work from the consortium. This paper is online in a preprint, is predictive microbial signatures, just looking at DNA. Can you actually predict what city you're from and how much in different cities, can you predict, some more than others? Zurich and New York City are among the more mysterious cities, if you will, at least in terms of prediction accuracy, but population and surface type do often give you some sort of signatures that can use that the classifier can anchor onto. And we can get up to about 88% accuracy. If you give me a sample, we can likely tell you where in the world it came from at the DNA level. So this is some ongoing work with Metasub, and I actually didn't put the preprint location on here, but if you Google Metasub bioarchive, you'll probably be the first hit, almost certainly. And so that's the bit of the background of the consortium. And other things we've been doing about the consortium is they're very dedicated researchers and creative. You can see here, Alina, 1 year, was detained a little while, and she said, well, can I sample this detention center as part of the project? So that became part of the sampling collection last year. Also, Naranhan Najrahan actually has got very creative with the people in labs you can see here in Singapore. So it's a very fun group, a lot of dedicated researchers. But in the past really few months, we've ramped up to do first a pilot sampling across these cities for looking at, historically we'd only looked at DNA, but now we're extracting DNA and RNA from every single sample to look at what's present. These are the first cities that started. And as of today, there's about 59 cities sampling about 10,000 locations across the world, as you can see on that map. So beginning this virus tracking to see what can we find in the different environments. So in particular, you can see, for example, this is a picture in Seattle, Washington. You can see here's David Denko from the lab sampling in Penn Station. You can see here in Alexanderplatz in Berlin. This is in Buenos Aires, the sampling location, some of the areas. This is in Daegu in South Korea, where there was a really big outbreak there a few weeks ago. We were sampling there, also in Brazil and Sao Paulo, and then also in Tokyo, Japan, among other places. This gives you ideas of, as the sampling's happening, there's an online app where you can see the geotag and the picture of the location. So one interesting bit of data of what we've seen so far from the shotgun sequencing of public surfaces is about 10% of the RNA is human, about 1 to 2% of it's viral, and a good amount's bacteria. But if you rank the viruses that we've just been seeing, and this is very new data, a couple days old, we so far have not seen coronavirus, which is a bit of good news, but we've seen influenza, we've seen pneumonoviruses, we've seen other sort of phage viruses. There are a lot of viruses to be found all around us. Some of these things are not spread. This is just the average abundance across the first 14 samples we sequenced. And you can see, number of reads, we're seeing a good number of reads and even coverage across the genome. So this is very new data, but it's interesting to see what else is out there in the world around us. And we just had another 60 coming off the sequencer today. So we'll share this data within the Metasub consortium, and anyone on this call is welcome to join the calls or hop on and play with the data as well. So that's a bit of shotgun sequencing services. Two other updates I want to give you quickly about what we're doing is ways to make this faster. So in particular, we've been working on a protocol called LAMP, which is a loop-mediated isothermal amplification. The way it works is every time a polymerase incorporates a base, the pH starts to drop in the solution. So as the reaction proceeds, it'll drop the pH and change the color. So it's basically just a pH test. And so using a synthetic RNA, you can see here at 0 minutes, it's all pink. Once it's at 20 minutes, you can see the color starts to change, especially for high viral abundance. And then once it's finished at 30 or 40 minutes, we've gotten it down to 30. We can see between 10 to 100 viral copies of what we can detect with this reaction. So we're excited about this because we've gotten it working as a rapid test in environmental samples, and we can confirm these negative detection of coronavirus or SARS-CoV-2. It works though for the clinical positives, and also the negative controls, it also works. So this is building off some work that was first testing it in China that showed that it was possible, and we've now done this on over 200 samples, and it looks really good, about 94% sensitivity and about 99% specificity. So we're very excited about this as a maybe more rapid test that we just got the IRB approved yesterday. We're going to be deploying this at the hospital here in New York City, hopefully by Friday, actually. So that's not everything we're doing. I also have kind of a dream slide of if we could do everything for every sample, which we can't always do because we're limited in material, but we're doing LAMP, we're also doing qRT-PCR and ribodepleted and sometimes poly-RNA-seq. We've begun doing some direct RNA sequencing and MinION work, and also doing look at capture panels with Twist, for example, with these samples. So this is kind of my dream slide. We're doing mostly these top 4, and sometimes doing a little bit on the bottom when we have enough RNA from different clinical samples. So speaking of what we've seen for clinical samples so far is if you do a ribodepleted RNA-seq, this is a coverage plot of what we see. So you can actually get really good coverage of the whole genome. And we're seeing anywhere from 60 to 70% of the virus from nasopharyngeal swabs to as much lower, like 0.1% or lower. It really depends on the sample. Actually, I have a slide of the correlation between the two, which looks so far pretty good, which I'll show you in a second. But we've been able to get consensus alignment. And as most of you probably have been doing, we've been uploading all of our data into Nexttrain. So you can see some of our samples down here in red. This is actually from a plot from a couple weeks ago. But we begun uploading all the samples here, which is I recommend. If you upload them to GISAD, then Nexttrain pulls that data from there. I highly recommend this site. If you haven't been there, it's probably one of the best sites to look at. You can see it's here. I think my time is up almost at 10 minutes, but if you haven't been to the site, it does a really great video of the sort of history of the phylogeny and distribution of the samples around the world. So so GISAid is essentially where people have been depositing their data. So we're putting our data. If you haven't already, any of you doing sequencing, even if you're doing targeted sequencing or shotgun sequencing for metatranscriptomics, you can do the alignments and call variants and then upload the FASTA file to GISAid. It's a really great resource that, at least as far as I know, is the most centralized location for all the viral sequences. So That's where we're putting all our data, and I recommend it everyone. So again, if you haven't seen this site, I presume most of you probably have, but if you haven't, it's really one of the more beautiful illustrations of when and how and where different viral strains have moved around the world. And also at the bottom, you can see where are the different phylogenetic distances, how are they mapping in terms of mutational frequency across the different strains that have been sequenced. So really extraordinary website. It was built by Trevor Bedford and his colleagues, and it's really fun to work with as well. So that's a quick update there. And then the last comments are we're trying to get set up Popup Labs across New York City to make it very easy to do this LAMP assay in the next basically couple of weeks. We're getting this set up hopefully soon. I think Danielle Bezin's on the call. We've done this before at other meetings where we just swab people's phones and sequence it and tell them what's there. It's just, in this case, it's for fun, but now we're trying to do this more for clinical purposes for testing people. And the idea of setting up Popup Labs all over New York City, we're working with the city council and also the mayor's office to begin to set this up, which we're working with right now with this LAMP assay, which we're submitting for FDA approval hopefully very shortly. So the long-term dream would be to not just look at the LAMP positive samples in RNA sequencing, but look at genetic variation, differential expression in the host cells that are there, look at epitope diversity, host immune cell diversity, look at changes in RNA function, and also anything else that's there. This is kind of my dream slide of everything we can do on the computational side with these samples from a host and microbial perspective. And then I'll close with a new initiative that just started on Monday, which is a host genetics initiative. So it's open to anyone to sign up. It's basically a place where people are looking at host susceptibility for sort of people that have high and low viral titers and sharing data from genome sequencing to array data to functional genomics data as well. That's my last plug for things. So stay tuned. Thanks to everyone. Thanks to Gunnar for the invitation and everyone else on the call. Thanks to especially the clinical collaborators and everyone in the Mason Lab. And thank you very much for your time. And also thank you, Krista, Ben, and the councilman for helping across setting up all the sampling around the world. And that's it.